Welcome everyone today. We're exploring a vital concept in ecology, population size estimation. Whether studying zebras on the savannah or snails in a garden, ecologists must be able to estimate population sizes to understand biodiversity, monitor conservation, and assess habitat health. Scientists use a range of techniques, each suited to specific organisms and environments. Let's begin with a real-life research scenario. A group of ecology students wants to estimate the population of insects and hedgehogs in a woodland. They use different methods, including quadrat sampling and line transects, and collect data at set intervals. This scenario introduces us to key principles of population sampling. Why not just count every single organism? Because it's impractical, especially for large or mobile populations. That's why we rely on estimation methods. Let's explore them. So, what exactly is population size? It's the total number of individuals of a species in a specific area at a given time. It fluctuates due to natality, birth rate, how many individuals are added through reproduction, mortality, death rate, how many are lost, immigration, newcomers entering the population, emigration, individuals leaving the population. Understanding these factors helps us grasp not just population size, but its dynamics over time. There are several methods ecologists use: direct count aerial photography, quadrat method line, transect method, belt transect method, capture, mark, recapture. Let's look at each in detail and when they're best applied. Direct count is the simplest method, but not always the most practical. Best for large visible organisms like elephants, lions, or birds in open landscapes. Procedure: physically count each individual in the area. While straightforward, it's time-consuming, labor-intensive, and often unrealistic for elusive or small species, or massive populations. Here's a high-tech approach: aerial photography, best for large animal herds like zebras or wildebeests in open areas. Procedure: use drones or aircraft to take photographs of the area. Count the visible individuals from images. This method is non-invasive and covers large areas quickly, but it requires specialized equipment and skilled analysis. Now let's break down one of the most widely used techniques: quadrat sampling. Best for plants and slow-moving animals like insects or snails. Procedure: place a square frame, a quadrat, randomly within the study area. Count all individuals of the species within the quadrat. Repeat this in several spots. Calculate the average number per quadrat. Multiply that average by the total area to estimate the population. But this method depends on key assumptions. Organisms are evenly or randomly distributed. The sampled area reflects the larger habitat. There's no major movement in or out during sampling. Individuals are easily identifiable and countable. If these conditions are met, the quadrat method provides reliable estimates. Next up, the line transect method. Best for organisms along a gradient, like plants on a hillside or species along a waterline. Procedure: stretch a string or tape a transect along the study path. Count individuals that touch or fall directly under the line. This method is efficient for spotting changes in species distribution over distance or terrain. Think of this as a wider version of the line transect, the belt transect method, best for plants and animals in a defined strip area. Procedure: set two parallel lines a fixed distance apart, creating a belt-shaped study zone. Count all organisms within the belt. Though more time-consuming, it provides richer data than a single line, especially useful when studying biodiversity across microhabitats. Now let's explore one of the most widely used techniques for mobile animals: the capture, mark, recapture method. Best for elusive or fast-moving species like rabbits, mice, or birds, organisms that can't be counted easily in one spot. Here's how it works. First, capture a sample of animals and mark them safely. Release them back into the population. After some time, capture a second sample. Count how many of these are marked individuals. Finally, apply a simple mathematical formula to estimate the total population. Let's look at that formula next. P. We use this equation. P as n1 times n2 divided by n3, where n1 is the number captured and marked in the first round, n2 is the total caught in the second round, n3 is the number of marked individuals recaptured. The logic here is simple. The proportion of marked individuals in the second sample reflects their proportion in the entire population. But for this to work, certain assumptions must hold true. Let's break down the critical assumptions. Closed population, no immigration or emigration between captures. If animals move in or out, the estimate becomes inaccurate. Random mixing, marked and unmarked individuals must mix evenly. If marked animals avoid traps or form separate groups, your data skews. No effect from marking. The mark shouldn't make the animal more visible to predators or alter behavior. If fewer marked animals survive, the population seems falsely large. Violating these can distort your results significantly. When using this method, ethical and scientific responsibility go hand in hand. Ethical considerations: avoid harming animals while marking. Minimize stress during capture, handling, and release. Scientific considerations: use marks that last but don't affect survival or behavior. Allow enough time between captures for proper mixing in the population. These best practices ensure your estimates are accurate, reproducible, and animal-friendly. Let's recap. Across this lesson, we've explored 6 different methods for estimating population size: direct count aerial photography, quadrat, sampling line, transects, belt, transects, capture, mark, recapture. Each method is tailored to specific environments and organisms. Whether you're surveying plants on a hillside or rabbits in a field, the right method makes all the difference. Question for you: which method would you choose to estimate the number of butterflies in a meadow, and why? Drop your thoughts in the comments and let's discuss. Thanks for watching. Like, share, and subscribe for more ecosystem